So you see this drawing, right? Micrograph is given and student is asked to draw plan drawing. And this is a result drawing. So the student has actually drawn individual cells. Okay, the drawing is actually quite accurate, but it's individual cell. So this is a wrong drawing. It's not plan drawing. It's a detailed drawing. Okay, so therefore it's wrong. This drawing is totally wrong. Okay, so I'm very surprised to see this drawing in the answer, because plan drawing means you outline the tissue layers. You don't draw any cell at all. Okay, now this one here, drawing is a bit small. You see the space is very big, so you need to try to occupy more than half the space given, better still 1/4 of the space, so it's a bit small, right? Other than that, I have to make it smaller so you can see, so they can compare between these two. So is the drawing accurate? I don't think the drawing is accurate, because how come my micrograph doesn't have got this part, but this student's drawing got this part? Where does this part come from? Question mark. Number two, I've got how many vascular bundle here? 1, 2, 3, 4, 5, 6, 7. I'm expecting 7 vascular bundle. How come here don't have 7? Why only 4? So therefore, this is not an accurate representation of this micrograph. So this drawing is again not correct. Okay, not correct at all. This one, really. So you see micrograph, there's so many tissue layers that you can draw, but this student only draw the vascular bundle, and the vascular bundle is also wrong. It's also wrong. Okay, and the labeling of the vascular also is wrong, because label has, if you use a bracket, the bracket must touch the object that you want to label. So again, this is wrong. Last one, number 1, how come these 2 circles are so nicely round? When you make a biological drawing, you need to use free hand to draw. You cannot use any instrument to help you to draw either the straight line or the circle, cannot, because nothing is exactly round or exactly straight in a living organism one. So you cannot use a ruler or don't know what is using. Okay. And then I can see that the student is trying to draw as accurate as possible for the xylem. So the orientation is almost okay, but still some improvement needs to be made. Okay, because my xylem is actually this one here. Now for your information, when you draw, right, you don't have to draw until you follow individual cells like this, like this, like this, going up, down, like this. No. You don't have to draw like this, okay? Because it's taking such a long time. You just overall a smooth line to cover almost that tissue like this. Okay, is already enough. You don't have to one by one the cell show, this is a cell wall going in, going out. No need. If you draw like this, right, you take a long time, right? Like this is enough already. So you look at the shape of this outline and this one, you can see that slightly is different, especially this part here should be bigger, but it becomes so small. So accuracy is out, thickness is out. Can you see how thick is this one here in the actuals, in the micrograph? How thick is this one versus this one is out, totally out, okay? And then the location, this is totally out also. Are you telling me that if this is cambium, if you're telling me this cambium, then this cambium is everywhere, is it? Everywhere in this space here, right? So this is wrong again. So please pay attention to detail and try to do correction and submit it again and make sure that it's accurate. Okay. This one is empty, so I don't know why the student didn't do this question. Why didn't do this question? This one has got answer one. Okay, this one is not done also. Okay, you need to answer the question and then you need to refer to the mark scheme and mark your answer and do the correction if it's not correct, or do the correction, you got additional points to add in and then submit. So that's what you need to do in order, then I will give you the full marks. If you submit something like this, I will minus 1 mark. That means 2 marks for every submission, minus 1 mark for did not do did not do and mark and check the answer. You do it, you didn't mark and check the answer, also is the same. -1 mark. Okay, because incomplete. Okay. Right. So this is for the first one. Second one. This one here, let us look at the drawing. The drawing is more, this is plan drawing at least. So this is, at least is a plan drawing. And we want to check the accuracy of drawing. Is accurate or not? It's not very accurate. You see, this is the first vascular bundle, right? Okay. Second vascular bundle, if I draw a vertical line, it should be located somewhere here. So these two are too close to each other. Accuracy is out. Okay, accuracy is out. This is a vascular bundle here because there's cells surrounding it. So for vascular bundle, for most of you, there are more details that you need to show. Because vascular bundle inside got xylem and phloem. So where's my xylem and phloem? It's not shown here. So you have to draw more detail than that. So first, you can see from the diagram that, am I recording? Yes. Okay. Now this diagram is not very clear. The previous one is clearer. So I use the previous one, okay? Okay, for this one, I make it bigger. So when you draw the vascular bundle, say for this one here, for example, I will draw this to represent the cells that surround the vascular bundle. You see these big fat cells here? They are the bundle sheath layer. So bundle sheath is part of vascular bundle. So this is where you need to know what are the layers, how to label them. Then these smaller cells are the xylem and phloem. And you can see they're obviously different one. Okay, so I'll base on the differences in the color to draw the borderline between the xylem and phloem. Okay, so therefore on top is xylem, bottom is phloem, and this is the bundle sheath. So after you label all these three, so usually that's what I will do. So how I will label it, I will label it this way. From the outermost, okay, and the uppermost to the lowermost, from outer to inner, upper to lower. So I use a horizontal line, and now I cannot because I got some labeling here and there. So I'll label this as vascular bundle, no. Bundle sheath. Bundle sheath is actually the name of the tissue, bundle sheath. I cannot write cell because it's I'm drawing tissue layers, right? And then this is xylem. And then this is phloem. So after having written these three, I will do a bracket like this. So ignore the spongy mesophyll, and I will give a name to this, which is called the vascular bundle. So this is what quite a lot of students didn't put when you draw your diagram for the stem and the roots. So in the stem, remember there are a lot of vascular bundle. You're supposed to draw one and label. So quite a number of students didn't label vascular bundle. So don't forget to label vascular bundle. So you can label it this way instead of you putting a bracket here and try to label it. Okay, because sometimes in the whole leaf, you only got one vascular bundle. So when you put a bracket, how are you going to put the line for the rest of them? Are you going to put the line here like this? If you can, then good. If you cannot, then how? So I will do it this way. Okay, bundle sheath, xylem and phloem, and I label it vascular bundle. Okay. So accuracy is out. For upper epidermis, in this case, you do not need to use the bracket because it is understood that this space enclosed by the upper and lower line is a layer. So this layer is upper epidermis. Therefore, you do not need to use a bracket again. Same as for here, you don't have to use bracket. Just point to this space and label it as, what S spaces is this? Okay, label it as palisade mesophyll, full stop. And then don't need the bracket. Point to this space and label it as spongy mesophyll. Understood? Because the border line is upper line, lower line. No need the bracket. Okay, unless you got a lot of other features, you have to point, oh, exactly this one here. No, no need. So no need bracket again for upper or lower epidermis. Okay. Next, this drawing is, let me put 50 so you can see the whole thing. Is this drawing accurate or not? So we count the number first. In exam, the examiner will count the total number 1, 2, 3, 4, 5, 6, 7. So 1, 2, 3, 4, 5, 6. How come my 7 here is missing? If you can see here, you have to draw. Okay, number is out, so -1 mark. Now the shape. The shape is also out. You see this shape is out. Why? You look at the actual shape. If I outline the shape, it's not the shape that the drawing has drawn. It's actually like this one. It's not like this. So the shape is out. This one also same thing. You outline the shape, you follow the shape and see. So this shape is out also. So -1 mark. So accuracy is very important. And then how come the cambium suddenly so thick one? How come the cambium is so thick? Cambium, you look into the xylem vessel. The part that is lightest in color. Again, this diagram is not clear. Let us use this diagram. Okay. How to see the cambium? You look into the vascular bundle and you look for the region with the lightest color. Why? Because the cells are meristematic cell. That means a cell that at the stage of cell division. So there's no thickening of the cell wall. There's no differentiation, specialization. So the cell is very thin in the cell wall and the smallest because it's not elongated. So usually it is brick shape. You know the brick that you use to build the wall, the brick shape. So it's usually brick shape with a thin wall and thin flat cell like this. Okay, and rectangular shape and very light in color. So therefore, when I see this, for example, I will know that this part is the cambium. And then how thick is the cambium? Just look at the white color region. So the thickness of cambium, I will draw in my plan drawing roughly based on this micrograph, roughly. But you're given a slide, it's not roughly already. When you're given a slide, you have to zoom into this one, means use a higher magnification, zoom in to look into here to see exactly where is cambium, and then you draw into your plan drawing. Because in a microscope, you can zoom in and zoom out. That means you can use high power and low power to check the detail and draw accurately. But now you cannot zoom in, so you just make use of whatever is here. So this cambium here will be slightly thicker than previous one. All right, so this is the thickness of the cambium. Okay, so sometimes in an older plant, you see that the cambium will also stretch to the space in between the vascular bundle here. So you have to follow this one. You see the shape of the cambium cell is rectangular. This is not rectangular, so this is not cambium. It's already elongated. Okay, so this one, which is also a cambium, which is found in between the vascular bundle, we call it interfascicular cambium. Okay, so I've given you a reference where you can check and find out the name of the labeling also. So you need to read. The references I've given to you is very good, or else I will not give you. So spend time reading it so that you learn better. Don't be lazy, No lazy. This one out. Okay, you have to do correction. This one out. Have to do correction. Okay, let's look at this one. All right, so this drawing okay or not? Let's put 50.
Teacher?
Yes?
Daniel told me to tell you that he's having internet problems.
Oh, okay. Thank you. All right. So this drawing is not accurate. This drawing is not accurate. Okay. But there are certain layers that are correct, like epidermis is correct. There's colon-chyma, of course, it's already labeled here, okay? Colon-chyma, and then parenchyma. Now, this is wrong. This line is wrong. This line is wrong because parenchyma will stretch all the way to here. Because don't forget, cortex will stretch from epidermis all the way to the cambium region in the stem. Okay, but it can be made out of different cells. So you've got different tissue layer like collenchyma and parenchyma. So you shouldn't have this line here. This line is wrong, out. Okay, then there's a pith. This is wrong. This is wrong. All labeling must be written outside the drawing so that you do not mix the line of the drawing and the line of your labeling and the words of the labeling. So this labeling must be done outside your drawing, outside the border of your drawing, cannot be inside. Of course, this one is a diagram, so they can do whatever they want, but biological drawing must follow the rules, okay? So again, wrong. Here, correct or not? Yes, this is correct. This is correct. Huh? Why? It's because the student has outlined the tissue layers correctly. All right, so what are the outline that correct? So you see this red color layer of cells, they are all the sclerenchyma cells. So therefore, all the cells where the wall has been stained red color because the wall got lignin, so she has drawn it all together as the same layer. This is labeled sclerenchyma, but what happened is that, how come there's no labeling one? Okay, every structure you draw, you need to label one or no, else I wouldn't know what is it. Okay, so this drawing is correct, but there's no labeling. So this is sclerenchyma, please label. So next week, you need to submit all your corrections for all your drawing. Okay, I don't care. Every one of you, whether correct or wrong, you must submit again. So when you do correction, you use either a red color or blue color pen, for example, for her case here, on top of her drawing, she will add in whatever labels that she has not labeled. Okay, make sure it's root line. So use red color or blue color pen and you write in it. So I know that your original one is in pencil. And then your correction is colored version. Then I can see. Then I know, okay, this one is correction. Let's say if your drawing is wrong, then you have to draw again. So you use a pen to draw it, you don't use pencil, then I know that that is your correction. If a minor mistake, you can draw inside original drawing. Let's say, for example, oh, I forgot to draw something here. You draw something here, inside here. Use color pen. So that's a correction you need to do. Pass up next week, Monday. Other than that, the tissue is correct. Yes. So tissue is correct. No, it's wrong. Tissue is wrong. Come back to here. Now, xylem and phloem, where's the borderline, is actually here. It's very obvious. Can you see? These two parts here, they are very different in terms of color and texture. And these cells here have got lignified wall, but these cells are totally like normal parenchyma kind of cell. So this part here, which looks like normal parenchyma kind of cell, is the phloem. The rest of them here is considered as the xylem. Okay, and of course, this whole thing here, you can label this as bundle sheath. So you can label this as bundle sheath. Then you can draw this as separately. Hang on, this is not correct. This is not correct, maybe. So I will see the cell wall is thickened where? See, okay, the cell wall is thickened here, it's outside the vascular bundle. So I label this separately as the sclerenchyma. So I'll be having a lot of sclerenchyma here, see, not at the side there. The bigger cells the bigger cells with the lignified wall. Okay, lignified wall only I will outline, okay? Right. What about here? Oh, this one lignified, you see, not? So I label sclerenchyma. So therefore, you need to know how to identify the cell based on its structure also when you draw plan drawing. This stem is very interesting, you see, not got stoma one. Okay, this stem got stoma. Is this stoma? Looks like it. Looks like gut cells here. See, this cell is different. Look like gut cell, and then got substomatal air space. So very interesting, this one. Okay. Next structure. This one finished already. Yeah, I don't know why in previous one didn't scan this page. You see, not this page here. Is this drawing accurate? It looks quite accurate to me. Let's make it smaller so we can check, okay? Accuracy, you can see that, yeah, quite accurate. You know, the orientation of this vascular bundle is quite accurate. The size is also quite accurate. Epidermis, endodermis thickness is quite okay. So after endodermis, now this layer with the Casparian strip, you can see quite clearly, right? So this layer here is the endodermis. Immediately the cells below the endodermis, this layer of cell here, okay, is the pericycle. So the pericycle seems to be thicker than the endodermis. So her drawing is quite accurate. You can see now, pericycle thicker than endodermis. So that could be one marking point in exam. Accuracy of drawing is marked in this way. Okay. Then vascular bundle got cambium or not got, but the location of cambium is wrong. Okay, the location of cambium is wrong already. So let's go back and check the clearer drawing here, clearer micrograph. Now, cambium, you look for the smallest cells are located here, flat and rectangular, right? So this will be the cambium. Here is not flat, can you see not? So there's no cambium at this edge here one. There's no cambium here. So you don't draw cambium. See, cambium for this one is even clearer. You can see the cells are not. Can you see the cell is quite flat and brick shape? So this is the cambium. Okay, so the cambium will be between xylem and phloem. Outer region here don't have cambium. Right, so just now that drawing is not so accurate. So therefore, that's where you need to know how to analyze the structure of the cells in order for you to determine whether this is cambium or not when you draw the plan drawing. Okay, so this one, make it smaller so you can see. Is this drawing accurate? I think this drawing got some problem. I do not know whether this student, do you actually copy seniors mark scheme? Seniors mark scheme, I take seniors drawing, right, to show the student, to show some mistake, and then it looks like this drawing is similar to senior mark scheme because senior draw S spaces, and this drawing also got S spaces. My goodness. Do you know why spongy mesophyll is called spongy mesophyll tissues? Because there's a lot of S spaces. So this whole tissue with the cell and a lot of air spaces, the whole tissue is called the spongy mesophyll. So you don't draw the air spaces because this is not detailed drawing. And on top of that, air spaces is not a tissue. So why are you outlining air spaces when it's not a tissue? And then why are you drawing gut cell in a plan drawing? Is gut cell a tissue? So this is something you need to pay attention. You are drawing plan drawing. So no cell, no non-cell material. This one, the drawing is quite accurate, but actually it's not that accurate. Okay, but can be better because I see that the size of this one is quite big. And suddenly this size, how come is like half the size of that one? But the other one is quite okay. This one is too near already because there's a gap there. Can you see there's a gap, but then suddenly here got no gap. And there's a gap here as well. And suddenly there's no gap here. Right, but I can see that it's quite accurate. This student is trying to draw quite accurately. So when you draw, try to actually space out and follow again. Follow the diagram wrong already. All labeling must be outside the border, outermost border of the drawing. Outside. Cannot draw this line. Wrong. Where got this line one? Okay. The pith will start from the cambium all the way inwards. Okay, unless it's a hollow pith. Then you need to draw this line to show where is the hollow part. So this line is wrong. Okay, so do correction and pass up next week, the whole class. This drawing is quite accurate. I can see this is considered as quite a good drawing, because you can see the accuracy of the drawing. Okay, is the labeling correct or not? Let me check. Is the labeling correct? Xylem, phloem. How come vascular bundle is here now? Why suddenly vascular bundle? What are you labeling? Oh, this one. This one is not acceptable. Why? Because you got label line overlapping, label line crossing. Cannot. That's why I say you cannot put a bracket and then label some more. Okay. So if you want to label like this, you put xylem here and you put phloem here and you label xylem and phloem as vascular bundle. Okay. So this one is not acceptable because there's a crossing line of the label lines and the bracket line. Parenchyma, colon-chyma epidermis. Do I have colon-chyma there? Do I have a colored version? Colored version, then you can know whether it's colon-chyma or yes. Is this a colon-chyma? I'm not quite sure. Yeah, it is a colon-chyma because, so if you got a slide, you can actually zoom in and see that there are certain corners of the cell wall that is thickened. Can you see this thickened corner here? So the cell with the cell wall thickened at the corner, they are the colon-chyma cell because they got the cellulose thickening at the corners. So this entire layer that's green in color below the epidermis is considered as colon-chyma. All right. Then I've got this is the sclerenchyma. Okay, and then this is the vascular bundle with whatever thing inside there. Right. I will label this as parenchyma. All these cells are in the center here. I'll label as parenchyma. I can also label it as the no, I'll label it as parenchyma. Why? Because now you only see part of the stem, you know. If you see the whole stem, you'll notice that the vascular bundle is scattered, so there's no pith. Okay, there's no pith for this one, but you cannot see because you only see the outer part. Okay, so now the next one, example only for you all to learn. Okay, so how's the drawing like? Okay, not? Drawing is okay, labeling is okay, except for gut cell again. Why suddenly go and draw a cell? It's a plain drawing, so cannot draw gut cell. This one, my God. The micrograph only give you 1/4, slightly more than 1/4. How come suddenly your drawing can become the whole thing, man? Where the rest of the thing come from? Then your total vascular bundle also wrong. This is not biological drawing. This theory. You need to report what you see in this slide, this micrograph. So this drawing is totally out. So I don't know how the examiner is going to give mark for this one. This one, you see what happened? I only give you maybe a small part of the stem. You're going to draw the whole stem. How do you draw the whole stem? You don't have the diagram also. So this one, be careful. You only draw what you see or what is given to you. Okay. This one also not accurate. See, not a lot of things are missing here and there. This one, I give you 2 diagrams because I want you to be able to use this diagram for you to help you to see what is this part here made up of. Okay, so this is a monocot root. A monocot root. So what you need to distinguish is very easy. This layer here is the endodermis because you got this. Can you see this thick wall, which is the suberin of the endodermal cell? Right, and the shape of this cell is different from the shape outside here. See, the shape outside here are hexagonal, and this one is rectangular. So therefore, this is a different cell. So this is endodermis. The cell immediately below it, right? All these cells here, you can draw them as the layer for pericycle. So after you draw the pericycle, then you have to find the border line between xylem and phloem. Very easy, because all these cells where the wall are thickened with lignin. Now these air spaces are So this is not the xylem vessel, okay? How do I know? You look at the wall. You look at the wall, the border here, there's no pink color. So these are air spaces. All right? So I will roughly draw like this to indicate, right? I will outline the borderline between the wall that is thick and the wall that is thin, like this, roughly, yeah? Roughly. Okay? So then I finish my job already. This is phloem. This part here is xylem. Finish. Okay, and then plus my, what is this? Endodermis and pericycle. Finish. That's all. Of course, in here, you still have to draw the epidermis. Okay, and label this as cortex. Okay. So you see what happened? Part of the root become a whole root. This is wrong. So be very careful. Biological drawing means you report what is given to you, whether it's the slide or whether it is the micrograph. This drawing is okay. So just that, don't have to draw the border of the individual cell so clearly, or else you have to spend a lot of time to learn to draw. Now, what is the time given to draw a plan drawing like this? For me, within 1 minute, you must finish the whole drawing. Within 1 minute, any biological drawing given to you to draw, whether it's detail or whether it's a plan, within 1 minute, you should be able to finish the whole drawing. So that's the speed that you need to be able to achieve. If it's too long, that means you are not familiar with the tissue layer, you have to learn it up. Okay. So this also same thing. No need to draw like individual cell, go up, down, up, down. Go up, go down, go up, go down, go up, down. Then 1 minute cannot finish. Just draw a smooth line across. You just have to roughly show the, my line is out already. Roughly show the border shape. Okay, the shape of the border of the stem. Okay, so again, pith location is wrong. Always remember, pith is after the cambium and cortex is before the cambium. So that will distinguish between cortex and pith. This one, okay, what is? Again, pith cannot label inside your drawing, okay? Pith, you don't label like this. You must have a label line to actually label it. Drawing wise, I think this drawing is quite accurate, right? And then no need to draw endodermis. Usually we don't see endodermis and draw endodermis in the stem. Even though you learn the basic tissue, got epidermis, cortex, endodermis in the stem, we never label endodermis, okay? So no separate layer for endodermis. This one, same thing. This is when you try to be very, very accurate, no need. So the border line, no need to enclose the wall shape of individual cell wall. Okay, no need. Right? Other than that, I think the labeling, everything wise is quite okay. I think this is quite okay. Last one, I'll say, last one is wrong in the sense that, do you know why is this wrong? Because the student is drawing the individual air spaces. Whether this air spaces or xylem vessel element, you cannot draw because you're drawing plan drawing. So you don't draw cell, you don't draw air spaces. Okay. So the drawing is wrong because the student draw the air spaces. So cannot. Then of course, there's no cambium lah, right? There's no cambium. There's no cambium there, okay? So don't draw cambium there. Okay? So I have got a sample answer that I will post it for you to check the answer. Let me check. I show you now if I can take it out. So this is a plan drawing, right? Plan drawing sample. I've got 2 samples. This drawing is quite good. Are you able to see this one here that I'm showing now? Can Oh no, too small. Okay, Now this drawing is quite good. You can see it's quite accurate. Even the location, everything is quite accurate. Okay, so I'm going to give you this one in your exercise answer folder for you to double check. Okay. This is considered as very good drawing. All right, not very good. I'll say good lah, good. Okay, accurate drawing. This one also quite accurate. This one also quite accurate. Can you see where's the location of cambium? And then this one also, see? This one actually, the student is drawing. You only need to draw this one actually. You only need to draw this one. This one is for you, is a magnified version of this part so that it helps you to draw the detail. All right, so that's why the student, you see the student's drawing is like this here. Okay, so drawing the whole thing. So this is a very good drawing. So I want you to, you can take this as a sample for you to do your correction. I think what is wrong here? There's something that's wrong here. There's no pith in the roots. So that's wrong here. So when you do correction, please don't repeat the mistake of this student, okay? Student's drawing is just a sample. It may not be 100% correct. So you have to know which one is correct and wrong because I've mentioned it in class. No pith in the root. I never teach there's a pith in the root. Okay, so that's all for today. So this is a correction. Please do correction and pass up on next week, Monday. All right. So tomorrow I'll see you for the class test. I'll release it into the Dropbox 5 minutes before before 9 o'clock. Okay. So that's all for today. You stay safe and have a great day. Bye.